This is an amazing blue ball we live on. My name is Veronica Castaldo. Please take a journey with me while we discover some of the crucial elements that make animals the magnificent beings they are today. In this video, we will be speaking about an extraordinary specimen. It's the oldest creature known. Can you guess what it is? Then I'll be introducing you to a very special guest. Sit down, strap in, and enjoy your world. As you know, there are different ways to classify animals, but why are they classified in this way? Well, in the beginning, there were unicellular protists in the ocean that needed to group together in colonies in order to successfully filter feed and survive. Flash forward millions of years later, the unicellular protist has now begun living together as a multicellular heterotroph we like to call a sponge. No, not that kind of a sponge. Yeah, that's the one. Many of you know a sponge is an animal, but why? It doesn't move like all other animals we know. It doesn't prey on other living animals. But yet it is an animal, and up until recently, considered to be the most primitive and oldest animal ancestor. Sponges lack neurons, muscles, and movement. These structures are all present in ctenophores. Another unique fact about ctenophores are they are well-known predators that use sticky cells called colloblasts to capture their prey, which can be sponges, echinoderms, or cnidarians. They possess two anal pores, which shows a relation to later deuterostomes such as ourselves. There have been three influential fossils found to be ancient ctenophores. One in Germany dating to the late Devonian period, and two dating to the Cambrian era, one found in Canada and one in China. Due to these fossils and genome sequencing done by Dr. Leonard Moroz, ctenophores have been speculated as being the base of the phylogenetic tree. Let us now go over to the studio where our reporter is interviewing Dr. Leonard Moroz. Very recently, at the University of Florida's very own Whitney Lab, Dr. Leonard Moroz has discovered another mystery to the already baffling ancestral tree. It appears as though phylum Ctenophora, also known as comb jellies, were formerly grouped with other jellies in phylum Cnidaria, due to physical traits, has been genome sequenced at being older than sponges.
Well, yes, it actually seems now that we have more missing links in our ancestry.
Thank you, Dr. Moroz. You are absolutely right. This genome sequencing technology is amazing. Can you tell me a little more about it?
Absolutely. Genome sequencing is done by taking a little bit of DNA from an animal and running it to see if that gene is present or not in its genetic makeup. Genes aren't always expressed, and because of this, we can't tell if they're present just based on physical traits. We can show a gene we couldn't necessarily see, and the only proof we need to figure out if the genes are closely related and the location on the phylogenetic tree becomes more determined.
Why, that is very interesting, Doctor. Now, why is it that even though Ctenophora has neurons, as well as every other animal, after sponges, are they still being dated before sponges?
Well, there is data in fossils of comb jellies that backs up this assumption, as well as the gene I found that Ctenophora uses to make these neurons work. It's different from those genes that every other animal after sponges uses to make their neurons. As to why and how this is, we're still researching ourselves.
Well, there you have it, folks. From the doctor himself, how and why animals are classified by Dr. Leonard Moroz. Thank you for your time.